Our today's topic is cell cycle. First of all, we'll discuss what is cell cycle. Cell cycle is basically a sequential series of events, or it is a chain of events in which the cell first prepares for the division and later on actually divides. सेल साइकिल में सेल सबसे पहले तैयारी करती है डिवीजन की और उसके बाद एक्चुअल डिवीजन होता है। तो इस लिहाज से सेल साइकिल हम दो फेज में तकसीम करते हैं, दैट्स इंटरफेज एंड एम फेज। तो इस इंटरफेज में cell actually it prepares for the division and basically the actual division occurs in the M phase। तो यहाँ से इस पॉइंट से लेकर इस पॉइंट तक M phase है, ये M phase कहलाता है जिसमें एक्चुअल डिवीजन हो जाता है सेल का। एंड फ्रॉम दिस पॉइंट up to this point it is the interphase। So the cell cycle is divided into two subphases, that's the interphase and M phase। But we have to remember one point over here, that the total time of the cell cycle is divided into 95% and 5%। That is, the interphase or the preparatory phase, it consumes 95% of the cell's cycle's time, whereas the actual division or the M phase, it consumes 5% of the time of the cell cycle। In this interphase, the cell prepares for the division, whereas in M phase the actual division occurs। Earlier this interphase was called as the resting phase. Now we understand that in interphase the cell basically prepares for the division, that's why we nowadays call it as the preparatory stage। So we shall start with the interphase। The interphase is divided into three subphases, G1 phase, S phase and G2 phase। So this will be the first phase of the interphase, this would be the second phase of the interphase, and this would be the third subphase of the interphase। So we will start first with the G1 phase। G1 phase is the first phase of the cell cycle। So in G1 phase, basically what happens, the G1 phase, in G1 phase, first of all we will understand why the term or why the letter G is used। The G stands for gap, it also stands for growth। It means to say that this G1 phase, it is the first gap phase, because G stands for gap, it is the first gap phase between M phase and S phase, or it's also known as growth phase, because in this phase the cell undergoes growth। So over here, if you will focus over here, it is G1 phase। In G1 phase the cell is basically metabolically active, it remains metabolically active and it grows in size, that's why we call it as the G1 phase। After the G1 phase is over, the cell enters into the S phase। In the S phase a very important process occurs, that is what we call as DNA replication, we call that as DNA replication, or we call that as DNA duplication। It means the amount of DNA which is present in the cell, it becomes double। The amount of DNA present in the cell is represented by 2C, the C represents content। So once the DNA doubles, it becomes 4C, again I will repeat, the C represents content। So the DNA content doubles due to the process of DNA replication which occurs during the S phase। So it is very important to understand over here that the process of DNA replication occurs during the S phase of the interphase। In case of these animal cells, also centrioles are present and centrioles duplicate during this phase। Besides DNA replication, centriole duplication also occurs in S phase, but only in case of animal cells, as plant cells lack centrioles। But you have to remember over here a very important point, that is the number of chromosomes doesn't double, as the chromosomes are not seen in case of S phase, the chromosomes are seen only in these what we call as M phase। And furthermore we have to see over here that only the chromosome content doubles, and the only the genetic material doubles, the content of the genetic material doubles, whereas the chromosome number remains the same। For example, in case of human cells, the chromosome number is 23 pair, it will remain the same even on completion of the S phase, but the DNA content will double, it will go from 2C to 4C। After that's over, now we will go into the G2 phase. Again, the letter G2 has been used in the context that is it is the second growth phase between the S phase and the M phase. That's why the term G2 is used. In this G2 phase, furthermore, the cell growth continues, and very important process which occurs over here is the protein synthesis. Once the cell completes this these three subphases, we say that the cell is ready now to enter into the what we call as the M phase. But we will focus on one very important thing over here before we close up this lecture, that is G0 phase. If the cell doesn't need to divide from the G1 phase, it will go into the G0 phase. For example, I will give you some examples like heart cells in case of humans and nerve cells. They usually do not divide after birth, and as a result, the cells do not need to go into the G1 phase, rather they go into the G0 stage, which we also call as quiescent stage, because the cells remain in quiescence. They do not further divide. If the cells would further divide, they have to go into the G1 phase, then into the S phase, then into the G2 phase. But these cells usually do not divide, so they remain in the G0 phase, which we call as quiescent state. Sometimes some cells are called bake for division in some special cases. Then those cells have to break their quiescence, and then they again have to go into the G1 phase, and later on they go into S phase, G2 phase, and they complete the interphase. One last point I will discuss over here is that the time taken for the cell cycle varies from the cell to cell. For example, in case of human cells, you will see the time taken for cell cycle by a cell is 24 hours. But if you see in case of the yeast cells, they only take about 90 minutes to divide. So this is all about the interphase. In the next lecture we shall deal the mitosis.